All right, so my project is understanding the role of cardiac macrophages in early life cardiac tissue development and function. So I come from a cardiology lab at CU Anschutz, and while a lot of my lab is mostly wet lab work, I thought I could use this class as an opportunity to try and get some more experience doing RNA sequencing, because this is something I'd never done before. So I asked around the lab, and a PhD student who also has never done RNA sequencing asked if I could maybe look into some data set that he's been interested in but has been too afraid to actually get in and try and learn this process. So he studies the early development of the heart and the role of immune cells, specifically cardiac macrophages. And so during early development, there's all these changes occurring in the heart, and the immune system plays a huge role in that to get from a baby to an adult. And so he wanted to know if some of these genes have a role, an important role in early development. And these were all ones that he had found from previous papers and stuff. So I said I would try my best to see if I could find any of these genes. And he doesn't know beforehand if any of these genes have a key role. It's more of just information can we use to guide us to future experiments? How can we narrow it down a little bit? So some of the tools used, first of all, there was a paper published that had publicly available RNA sequencing data, and it was all published on the Gene Expression Omnibus, which is a website that has lots of high throughput gene expression data, and a lot of papers will go there to publish their RNA sequencing data for anyone to use publicly, which is great because it gives us the base file to use, because running that ourselves could have cost thousands and thousands of dollars. They don't give you all of the finalized data sets, but at least gives you the raw file, so if you want to run the whole analysis yourself, you can do that. So some of the other tools used, my main one was called usegalaxy.org, and it's an open source web-based platform for biomedical research, and they just want to try and make this research more accessible, reproducible, and transparent. And I actually hadn't heard of this website before, but they have huge grant. Every year they're getting between $1 and $2 million of funding per year to continue doing this. And they have tons of researchers who will help write tutorials, help keep the website going, and just try to make it a great resource for all kinds of scientists. And another resource I used was they have a training website with Galaxy, and there was a tutorial on how to run reference-based RNA sequencing analysis. So that was a huge help for me to learn just the basics of how RNA sequencing works, because I really knew nothing. So it was going through this whole tutorial with a practice data set first, plus just doing a lot of reading, watching videos on my own to just try and understand what's going on. I think I have a decent baseline, but really this has taught me that there's so much more I don't even know yet, but it's been a good practice. So there were a ton of tools I used. I'm just going to show a few of them. The first one I used was Falco. It's also called FastQC, and it just gives a quick quality control of the data set that I have, and it will tell you if there's any major issues in the quality of your data. And one of the tools I used to fix some of the issues, so this is all of the samples that I had, and then this is a summary table. It'll say, all right, are your basic statistics fine? Yep. Are your per tile sequence quality fine? Yep. Oh, there's issues with adapter content or the per sequence GC content. And then they also give you more graphs that tell you more details about what's wrong with these things. And one of the tools I use to help with quality control is cut adapt, and it would remove adapters from my data set, because that's just a process of how RNA sequencing is run. They have to put in these adapters so they can make these reads, and then later you have to go in and remove it yourself from the data. The next thing, so once I had my data cleaned up, we had to use star alignment. So you have to take this reference genome, which you can find them online. I was using human data, so I used a human reference genome. And it'll have, the whole human genome. This is something that gets regularly updated. And then it'll take your reads and try and find the sequence of, A, G, C, and T, adenine, guanine, thymine, cytosine, and it will try and line it up with the genome to figure out where all of these little reads that you have in your data set belong. Then you can use feature counts, because we're interested in looking at upregulated genes, which means genes that are expressed more often. So it can look at different genes and it will see in my reads, all right, do I have one read in that gene? Do I have 500 reads appearing in that gene? And we know that, so you'll take your control and your treated groups and compare them and see, in this treated group, this one gene has 500 reads while my control has one read, so you know that this gene is doing something important in your treated group. So next I use DE-seq2, and that'll help determine what genes are differentially expressed. So we want to know so there's thousands of genes and we don't know which ones are actually important. And once we have our data and we have our different genes, to know which ones are important, the first thing you'll do is look at the p-value, what's statistically significant or not. The other thing you'll look for is the log fold change, and that tells you if something's biologically significant, because sometimes two values can be significantly different from each other, but that doesn't mean they're actually it's something that's a big enough difference that we care about it, if that makes sense. Sometimes values can still, because we have thousands and thousands and millions of reads, and so two values that are close together can still appear significantly different from each other just because of how much data is backing them up. But in reality, when you look at that big picture, they're not that far from each other. So that's why we need both values to tell us which genes are differentially expressed and actually important to us. So some of my knowledge gained. One of the first things I wanted to do was know how similar my data was to what was published in the original paper to know if I actually did this right. And I knew going throughout the process I had some issues with my data set. But so this is the figure from the original paper, and it is shown I have two groups, the fibroblasts and macrophages versus macrophages, and then this group, cardiomyocytes, fibroblasts, macrophages versus macrophages. And they did tell us, how many upregulated genes they found in this group and how many upregulated genes they found in this group. And then I went through my results to see how many upregulated genes I ended up with. And while they're not extremely close to the original, you got to think that before you do this cleaning, run DE-seq2 to know the important genes, I had 57,000 genes, and then it got narrowed down to this 200 and 2,400. So I think when you think about where it came from to where I am now, I think that was fairly close, although I can still tell that I needed some work with my data set so I can get closer to what the original paper had. And another thing I wanted to do, the reason why this data is publicly available, why I had to redo this whole thing, is because this is all I got from the RNA sequencing data set. They didn't give me a list of all the upregulated genes. They didn't tell me more details. You have to get that yourself. So they're only going to publish the genes that they're interested in. So I thought, why don't I take a few of these? I'll just took the first two from each group and see if I can see them in my data set, so I know, all right, I must have done something right if we have some of the same upregulated genes. And so I picked 6 of them, and 4 of them did appear in my data sets, 2 of them did not. So I know there's some issues with my data set still, but I feel like I was on the right track at least to know that I had some of the same upregulated genes as the original paper. Some of my other knowledge gained. So going back to what the PhD student in my lab originally wanted, he wanted to know if these genes were present, were upregulated or not. And this is something we had no idea. It was totally an exploratory type of research. So we didn't have any kind of prediction. But these are genes that he had read in previous literature that he knew would be important, so he thought maybe they would. And how I set up this table was the first data set, which is the fibroblasts and macrophages, is it present or not? And the second data set, cardiomyocytes, fibroblasts, and macrophages, is it present or not? So none of these genes I found in my first data set, but I found 4 of them in the second data set. So now that we know these genes, this can help guide further experiments, because if we didn't know this, we'd have to test separate experiments with every single gene to try and see, is this important or not? But now we already know these are important. What further tests can we do to understand, what is their role in the process? What do we need to look more into? So I think I didn't get as far as I wanted. I also wanted to do gene ontology analysis, but I think overall for my first time, this was a pretty good start.